All right, guys, so this is to kind of catch you up on what we would have covered in class. So we're going to talk about the endomembrane system, and then we're also going to be talking about kind of the origin of eukaryotic cells. And this stuff should get plugged into your note guides for the eukaryotic cells. It'll basically be the last 2 pages. All right, so the endomembrane system. Well, so membranes within the eukaryotic cell that work together are known as the endomembrane system. So it's multiple organelles, and they work together. They're going to modify, process, ship all sorts of stuff around the cell. The most common stuff that they're shipping is proteins, but they ship other molecules as well. So take a minute, think of some adjectives that you could use to describe a membrane. Some things you might come up with are like a bubble or phospholipid bilayer. Remember that the membranes of like the cell membrane, the nuclear membrane, and then also all the membranes of the endomembrane system are made out of a phospholipid bilayer. If you don't remember what that is, you can think back to when we talked about origin of cells, and we talked about the lipids actually organizing themselves in this bilayer. You can always look it up as well to get an idea. They're also semi-permeable, which means some stuff can go in, some stuff cannot, and they're fluid. They are movable, okay? So the endomembrane system, they're all made of phospholipid bilayer. They include kind of these 7. The nuclear envelope, the rough ER, the smooth ER, remember ER stands for endoplasmic reticulum, transport vesicles, the Golgi, lysosomes, and secretory vesicles. So this is a nice picture of the endomembrane system. You can see here is the nuclear envelope. Here's the rough endoplasmic reticulum covered in ribosomes. Here's the smooth endoplasmic reticulum. Here's a little transport vesicle. Here's the Golgi into a secretory vesicle that is then being secreted outside the cell. Here's another diagram. This one is the one from your book, or I'm sorry, your notes, right? And prior to actually moving through the endomembrane system, a couple of things have to happen. One, we have to produce the mRNA from the DNA in the nucleus. Then the mRNA is going to leave the nucleus through the nuclear pore here, and it's going to get ready to synthesize a protein on a ribosome. Now, the ribosomes are either attached to the endoplasmic reticulum, which is kind of what we're going to think about right now, or they can be free-floating, and we're going to talk about that distinction in a little bit. So just to continue to remind you about those micrographs and where we would see some of this stuff, you can see here's a micrograph of the actual nuclear pore. There's the nucleus and there's the cytoplasm. And a couple of micrographs of the pore complexes, the surface of the nuclear envelope, and the nuclear lamina. Okay, so in your notes, you can see that there are 1, 2, 3, 4, 5 steps. This correlates with that first step. So using the RNA code, a protein is synthesized on the ribosome and transported in channels of the endoplasmic reticulum. Remember, as you do this, you can always pause and catch the information. So what is the endoplasmic reticulum? Well, its biggest function is to process proteins, build proteins, manufacture membranes, and the way that it's built is actually these connected membranes to the nuclear envelope, and it goes all throughout the cell. Some have more endoplasmic reticulum depending on their job, some have less. So you can see here you have these nice endoplasmic reticulum that are rough. They're covered in ribosomes, which means they're building proteins. You also have the smooth endoplasmic reticulum over here, which manufactures the actual lipids. Specifically, one of the biggest lipids that they're creating is the lipids for the membrane. Here is both an electron micrograph and just a picture of the difference between rough ER and smooth ER. You can see that they're identifying a smooth ER versus, say, a Golgi can be a little bit tricky unless you have a really good picture. Typically, the smooth ER is going to be attached to the rough ER, so it should be right next to it and be in kind of these pretty complex system of what looks like Golgi, but they're not. All right, so step 2, protein is packaged into transport vesicles, and they travel to the Golgi along the cytoskeleton track. So that's what's happening here. What do I mean by cytoskeleton track? Well, throughout the cell are microtubules and microfilaments, and those both make up the cytoskeleton. It's kind of like all of the pipes and rebar and things like that are in the walls. And instead of just floating, it's not like a bubble that floats away, vesicles that have information inside them, proteins or other molecules, are actually carried by what's called transport proteins. So these little proteins actually will travel along the microtubules and microfilaments to wherever they need to go. They are basically the highways of the cell. And we need to have that, or else the cell would just collapse on itself. It would not be able to actually transport information. Here's a nice image of actually seeing all those microtubules and microfilaments. All these little lines coming out of here are the cytoskeleton of the cell, and it is what everything is attached to and built on. There's a video clip here. Obviously, I'm not going to play it while I'm talking, but you can go into the Haiku page and watch this video clip. It just shows a nice little video, it's a cartoon, of vesicles actually traveling along the microtubules using those travel proteins, those transport proteins. So step 3 is that the vesicle fuses with the Golgi, and the protein is then modified as it passes through. So the Golgi is basically like the UPS or post office. It will finish the proteins, modify them as needed, sort them, tag them, and then ship them depending on where they need to go. So they ship their products in more vesicles. So vesicles come into the Golgi as transport vesicles, as you see here, and they leave the Golgi as secretory vesicles. All right. Step 4 is that those completed proteins are then packaged into those secretory vesicles for release from the cell, or if they're being used inside the cell, they can be stored or be moved into lysosomes to be digested. So you can see that that's happening here. And then really step 5 is that the secretory vesicle is excreted outside of the cell. Okay, and I didn't put that down, but I can. So these are just some different images of vesicles actually being transported through our system. Okay. There's a second video that shows this really, really nicely. I showed it in class, and I will probably show it at the beginning of our class just so that you can see it, and I'll talk you through how the different organelles work together. All right, quickly I want to talk about structure and function and how we can identify specialized cells through micrographs. So different cells have specialized shapes and specialized organelles to perform whatever their function might be, right? The small intestine cells are going to have different organelles needed than, say, the gland secreting cells or photosynthetic cells or neuron cells. They all have their own job, and they are quite specialized, and you can see that here. These are 4 different types of eukaryotic cells. They have all the characteristics of a eukaryotic cell, but they are unique to what their job is. So let me talk you through a couple of those. So what you can do is pause this, and I want you to think about what organelles can you identify. Think about the role of the organelles that you can see in this picture, and then think about what the major function is. So pause it and try that. Okay, so what you should have done is think kind of through these steps. Well, there's a nucleus present. There is no cell wall, which means this is an animal cell. And to go back, nucleus present means it's not a prokaryote, right? So yes, nucleus, no cell wall, so it has to be an animal cell. I see lots and lots of rough endoplasmic reticulum all through here, which means that because it's present and it dominates most of the cell, we have lots of protein production, and that protein is going to be secreted. When I said a little while ago that the endoplasmic reticulum ribosomes are made for creating proteins that are going to leave the cell, but then there's free ribosomes that float around, and they also make proteins, but those proteins are used inside the cell. In addition to rough ER, I see lots and lots of mitochondria. Those are the kind of these long skinny bits in here. Those mitochondria are going to be synthesizing the protein, they're going to be taking the synthesized protein and using it, right? So those proteins are going to be used, and a mitochondria produces energy. So in order to synthesize proteins, we need lots of energy. So we have lots of mitochondria for that. In addition, we have lots of secretory granules or vesicles, and that's what these black circles are. I'm going to guess they dyed the vesicles on the slide so we could see them better. And so because there's lots of secretory vesicles, lots of RER, lots of mitochondria, this is probably a cell that's specialized in secreting protein products, probably hormones or enzymes. And that is, in fact, exactly what this is. This is a pancreatic exocrine gland cell, and that's its whole job, is to secrete information. I want you to try another one on your own. So here is another cell. I want you to pause it and try to do the exact same thing with this one. What organelles do you see? What organelles dominate? What would then be the job of this cell? Okay? And then we'll talk about this one in class. All right, pause the video, take a break if you need to. I'm going to move into talking about the origin of eukaryotic cells. So the Earth is very old, 4.5 billion years old, and life didn't come around till probably 1 billion years after that. And for another 1 billion years after that, prokaryotes were the only thing on Earth. So where did some of this other stuff come from? Well, it started with cyanobacteria, and cyanobacteria is a type of prokaryote that has lots of infolding of its cell membrane, and it's actually able to do photosynthesis and release oxygen into the atmosphere. So oxygen started to accumulate around 2.5 billion years ago, and we're able to actually see this in the rocks. So you see these little red bands. Well, actually, those are evidence of oxygen because the iron that was prolific all over the Earth previous to eukaryotes coming started to rust. It started to bond with the oxygen and create rust. And so because this oxygen is beginning to accumulate, aerobic respiration was beginning to be possible. Now, eukaryotes were able to evolve from this. Nearly all eukaryotes are aerobic. They need free oxygen in order to do their metabolic processes. So they could not have evolved before at least some free oxygen was present. Oops, sorry, skipped forward a little bit. So there's two processes. There's infolding of the prokaryotic cell membranes, and then there's endosymbiosis. So infolding of the membranes. So over time, the internal microenvironments were created about 2 billion years ago, where these membranes would actually infold inside the prokaryotic cell, and the cell was able to increase its efficiency at actually being able to transport information and create what it needed to create. And so due to natural selection, it was selected for, and that infolding became more and more common until we see what we see today with the eukaryotic cell. So in addition, the nuclear envelope started to form at this point as well, and that's how we kind of moved into the proto-eukaryotic cells. Now, given what you know about infolding in the cell membrane as a phospholipid bilayer, why is the nuclear membrane a double membrane? Why does it have 2 layers of a bilayer like this? See it? Why would that be? Maybe think about that a little bit. So the next step is what we call endosymbiosis. So we have this early eukaryotic cell that has infolding of its cell membrane, and so it's very effective at doing its job. What's interesting is that some of the first early eukaryotes probably ate or engulfed an aerobic bacteria, right? An oxygen-using bacteria, but it didn't digest it. This is the origin of mitochondria. It's very interesting because by taking it in, the cell was able to then start to create its own energy. It didn't have to use other energy. And so this was incredibly beneficial for both, because now the mitochondria, or early aerobic bacteria, was able to actually use nutrients from the cell. It didn't have to find its own. And this original eukaryotic cell was able to use the energy from the mitochondria to continue to create its own energy. So it was mutually beneficial. We also see this with photosynthetic bacteria. So once you have this early eukaryotic cell with a mitochondria, it may have engulfed a photosynthetic bacteria as well. And by not digesting it, it was able to actually harness the energy from the sun and create a mutually beneficial relationship, leading to the origin of chloroplasts. So what I'd like you to do, there's been enough of me talking. This lecture is posted, and I would like you to take some time to go through the remaining couple of slides to write down the structural evidence, the genetic evidence, and the functional evidence for these 2 processes. Thanks so much for listening, and I will see you in class.